Mycobacterium tuberculosis is a pathogenic species of bacteria which infects the throat and lungs. To begin, once MTB is in the alveoli of the lungs, it comes into contact with a resident macrophage, where it will be recognized as a pathogen when MTB binds to surface protein receptors of that macrophage. The binding of the two is achieved by the mannose receptor on the macrophage cell surface. This is a C-type lectin receptor, which is composed of 4 main subunits: C-type lectin 4, 5, 6, and 7. In here you can see the binding site in which the mannose receptor's ligand, a cell wall component of MTB called manlam, will bind. Manlam is composed of lam, which in live MTB gets mannosylated, meaning a mannose cap is added to it. This creates the entire manlam ligand. Nevertheless, once manlam binds a specific signaling adapter molecule, GRB2 is able to recognize the binding motif on the mannose receptor and will also bind. Once GRB2 binds and is activated, the mannose receptor activates AC1, CDC42, and PAC1 signaling cascade pathway, which is needed for F-actin polymerization in the macrophage's membrane. This ultimately allows for the macrophage to configure itself to surround the MTB and engulf it, forming a phagosome. Generally speaking, macrophages are programmed to attack or engulf pathogens and consequently degrade them to kill the pathogen. However, what makes tuberculosis so unique is its ability to evade these mechanisms and survive. If, for example, dead MTB were to be phagocytosed, manlam would bind to the mannose receptors, activating an influx of the macrophage's cytosolic calcium signaling, as well as signaling calmodulin and their downstream effector calmodulin-dependent protein kinase 2. At the same time, Rat5 would be recruited to the early phagosome, which would allow for the recruitment and phosphorylation of human VSP15 and the activation of class 3 PI3 kinase VSP34. Both of these events will create a phosphoinosite 3 kinase HVSP34 cascade, which would ultimately generate phosphoinosite 3 proteins. Once phosphoinosite 3 proteins are produced, Rab5's effector protein EEA1 would be recruited. Since EEA1 is essential in tethering and fusing the membranes of the lysosome and phagosome, a phagolysosome will be created. EEA1 would cause various other proteins, such as cathepsin D, to be recruited, and would ultimately. However, this pathway is completely different when live MTB is phagocytosed. Once bound, SHP1, a protein triazine phosphatase, is recruited by a GRB2-mediated recruitment to the mannose receptor on the phagosome. This will downregulate IF and gamma-mediated nitric oxide production and IF and gamma-inducible gene expression. Also, once SHP1 is recruited, Rab7 recruitment is inhibited. Simultaneously, Rab5 is recruited, leading to the recruitment and phosphorylation of the regulatory subunit VSP15 and activation of the class 3 PI3 kinase VSP34. Since VSP15 and VSP34 contain a consensus SHP1 binding motif, all three interact, causing a conformational change in VSP34. This conformational change leads to the inhibition of phosphoinosite 3 proteins production, therefore inhibiting EEA1. Simultaneously, while binding to the MR, manlam inhibits the influx of cytosolic calcium, and therefore inhibits the entire calcium cascade. The inhibition of this cascade also leads to EEA1 inhibition. Due to all of these events, Mycobacterium tuberculosis is able to evade degradation and can now survive within the macrophage.